Hello, everyone, and welcome to today's live broadcast, Mutation Detection Simplified, from liquid biopsy in the clinical oncology to the detection of drug resistance associated mutations in virology, presented by Dr. Michel. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Vela Diagnostics. Vela Diagnostics is a worldwide supplier of integrated life sciences and diagnostic solutions that help provide customers with valuable molecular information. From scientists striving to make research advances to technicians reporting the information necessary to identify, monitor, and treat diseases, Vela Diagnostics is a trusted partner for research and clinical laboratories around the globe. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. We have two speakers for you today, Dr. Gerd Michel and Mr. Welebob. Speaking first today is Dr. Gerd Michel. Dr. Michel obtained his PhD from the University of Heidelberg, Germany. After 2 years in the private laboratory sector, he joined Abbott Laboratories as a founding member and was with Abbott Diagnostics for 18 years, rising to the post of Director of Medical and Scientific Development for Europe, Middle East, and Africa. He was responsible for OEM product development and the assessment of new technologies and had numerous product successes, including HCV, HIV, and cardiac troponin I tests that became state-of-the-art diagnostics worldwide. For over 7 years, he held the position of SCO at the Foundation of New Innovative Diagnostics, FIND, in Geneva. During this time, he focused on the discovery of novel biomarkers for use in point-of-care detection of infectious diseases such as TB and malaria, mainly funded by the Bill and Melinda Gates Foundation. He became Vice President of R&D and Chief Technology Officer at Vela Research in Singapore in 2013, focusing on molecular and personalized diagnostics. He holds fellowships from the University of Aarhus, Denmark, and the German Cancer Research Center. He has also held a number of advisory posts, for example, at the WHO and the OECD. He served as a member of the German National Blood Advisory Board of the Ministry of Health and is currently a member of several research funding committees, for example, at the Wellcome Trust in London and the Global Health Innovation Fund Tokyo, respectively. Please join me in welcoming Dr. Michel. I will now turn the presentation over to him.
Good morning, everybody. Thank you, Christy, for the introduction. I'm Gerd Michel. I'm talking to you today from Singapore. And Vela will have two presentations in this webcast. One will be on virology, the other one will be on oncology. So I'll start today with part one, entitled The Analysis of Drug Resistance Associated Mutations in HCV and HIV using Next-Gen Sequencing. At Vela, we have developed a core technology concept which is shown here. It's an integrated workflow for NGS, next-generation sequencing, and qPCR, real-time PCR. And the principle is shown here. The workflow starts with an automated sample preparation system which we developed in collaboration with Eppendorf in Germany. And the user has then two options after RNA or DNA has been prepared by the system. either to go into qPCR or into NGS. And these two pathways are shown schematically on this slide. After the assay has been completed, there will be a reporting system which is specific for these two pathways, and the software which is related to it has been developed by Vela itself, so it's proprietary software. In the end, the data generated can be linked to local laboratory information systems according to the laboratory of choice. The whole system takes about, on average, 2.5 hours for qPCR and 2 to 2.5 days for NGS. I'll come to this in a moment. It's fully automated, and the report is also automated, so you don't have much hands-on time, and it's a very flexible system which can be adapted to your local needs. Talking about NGS specifically today, we're talking about an NGS system which is out there for quite some time. It's been developed originally by Life Technologies, now Thermo Fisher. It's called Ion Torrent NGS, and the schematics are shown here. What it is, it's a system that detects ion flow based on binding of nucleotides. And these reactions happen in millions of cavities on this chip, which is shown here, the so-called 318 chip, which we use in our system. So what you record is every binding event released a proton, and this proton is measured and transferred into an electrical signal. So you have multiple signals at a single time, millions of reactions on the chip. The nucleotides on this chip are flowing sequentially over the ion semiconductor chip, so you have a large number of records at the same time. I'm not going into much technology detail because of time reasons. There's a lot of information you can obtain on the internet or also from Vela separately. The workflow we developed for NGS for HCV and HIV genotyping is shown here schematically. The different steps are shown in the upper panel. So we're starting in both cases, since these are RNA viruses, with RNA extraction, then the library preparation, template preparation, sequencing, and the data analysis and reporting. These are generic steps in this workflow. The instruments related to these, used for these different steps, are shown below. So the Sentosa SX101, I already mentioned, that's a robotic sample preparation system. Then we use additional systems for PCR, like the Verity cycler, and then followed by template preparation on the ST401, sequencing on the Sentosa SQ301, and then the Sentosa reporter will give you all the data in a comprehensive summary. The reports generated here are two reports. Typically, one is a quality report that gives you information about the validity and the quality of a single run that has been conducted, and the other one is a pathology report that gives you information about the clinical significance and clinical meaning of the results generated. Reagents are shown here. These are kitted reagents which are supplied by Vela as well, and the software is shown at the lowest level. We have our own software development, as I mentioned, and we're continuously adapting the software to the needs as the assay assays are being developed. One important thing for NGS, which is in contrast to single gene detection where quality control is well established, this is not the same thing for NGS. So we had to develop our own control concept here, and if you want to have more information, more detailed information, or refer to this one publication, which is cited in the headline by Chan and co-workers 2016 in the Journal of Applied Laboratory Medicine, where we published a lot of detail about the concepts, the control concept. So in short, what we have here is 15 cycles in a run. This is typical for these assays. We have a system control, which comprises of a high concentration plasmid DNA, and the system control is processed as a standalone sample itself. It's shown here as the first ring on the schematic drawing here. You can see it here. And then we have an extraction control, which is a low concentration plasmid DNA. And this extraction control is spiked into all samples, is processed through the whole circle of the assay. And SC and EC goes through extraction, library prep, template prep, and sequencing. So using this control concept, we are able to control for contamination, extraction failure, workflow failure, and the presence of inhibitors in samples. So let me proceed. Let me talk about HCV, NGS, the assay design, and the workflow we developed. And this is a schematic drawing of the HCV genomic organization. What we do here is post genotyping, which happens in the NS5B region, which is the RNA-dependent RNA polymerase of the virus. And in addition to this, we're doing drug resistance testing or resistance testing or testing for resistance-associated variants, more precisely, in the NS5A and the NS3 regions. So what you get in one run is information on the genotype and also on the resistance-associated variants. We are using NS5B for genotyping because it has been shown convincingly in the literature that this has several advantages over the usually used 5' UTR region, which has been used traditionally for HCV genotyping based on line probe assays. And we're not using this region anymore because of several reasons. One is a reason which is coming up recently only is HCV recombinant viruses. Why do we need still HCV genotyping? There has been a lot of discussion about the introduction of new drugs. We have come a long way in HCV treatment from the discovery of the virus back in '89, and then the first introduction of therapy based on interferon subcutaneous injections 3 times a week, very cumbersome treatment regimen. Over time, the maximum response rate that was achieved in genotype 1 was around 60%. Initially, it was only 10%. So with the introduction of the direct-acting antiviral agents, first in 2007, the first generation, and then followed by the second generation in 2013, the picture has changed dramatically. Now we're seeing response rates, even cure rates in HCV of more than 90%. So why do we still need to do genotyping in RAVs? Well, as you will see in a moment, the treatment recommendations are still based on genotype selection. And also, RAVs exist in several regions of the virus and may cause some problems in patients, especially in those who have failed already interferon therapy. There are other reasons of failure. There is increasing numbers of baseline resistance, which is now being elucidated with the advent of the new technologies. And most problematic is typically patients which suffer from advanced liver disease, meaning cirrhosis, and in particular, decompensated cirrhosis. There's a strong opinion about experts that you will also see more RAVs coming up over time due to drug selection pressure. So what these new technologies also give you in more detail is more precise subtyping, which has been an issue with the line probe assays, which are on the market for a long time, which do not give you a very precise subtyping. So there's little information about the significance of subtypes, especially with the exception of subtype HCV1A and 1B, which are well established, but the other subtypes are not well understood. The question is there a clinical significance? And we do not know this. Only with the new technologies we can now take a closer look at it. And last but not least, importantly, there is in many patients we see mixed infections which are not readily detectable by line probing assays. And that is important because mixed infections may change the regimen for treatment. So as I said, genotyping is still important, as shown here. This is a summary of recommendations by WHO 2016. And what's shown here is typically a combination of drugs. Usually what we do today is drug combination treatment between of 1 to 3 drugs, typically, of 4 classes. And what's shown here is also the duration of therapy and which is classified by genotype, as you can see. So you need to know the genotype to determine the correct duration of therapy and the selection of drugs. So genotype remains an important factor in treatment, especially because the current treatments are dramatically expensive, still everywhere in the world, and that makes it more important to have a precise selection of the drugs up front. So this is a summary of the genotyping assay we developed on NGS. The major characteristics are shown here. 15 samples can be run can be used in one run. We have one system control, as I explained. The analytical sensitivity for genotypes 1 to 4 is 1,000 international units per milliliter. For 2,000, for genotypes 5 and 6 is 2,000, simply for the fact that we didn't have enough samples to validate, especially genotype 5, which is a very rare genotype, mainly present in Africa. So we're still working on it, and that may change also down to 1,000 international units at a later stage. We haven't seen any cross-reactivity with a number of viruses and human genomic DNA. Reproducibility in our ISO-certified assay development scheme was 99%. We're using EDTA plasma or other anticoagulants and also serum, and the total volume is 530 microliters. We're also using the UDG system, which is not a novel innovation, a novel invention, but it's been used widely in this area for the control of unwanted PCR amplicants that could interfere with the assay. Total assay time for HCV is 2 days. The hands-on time is minimum, only 2.5 hours. And the sequence data analysis is fully automated. And most recently, we have added another option to the software, in that we can now export also the FASTA files, which gives you additional advantages and options for your own data analysis if you do not want, if you want to use your own system. Now, a couple of data on the performance of the assay. This is an early study which was presented by Professor Coy from the Singapore University Hospital in 2015 at the American Liver Meeting. They looked at 150 samples and used the Lamprobe assay, which was established in their laboratory, the Versant HCV from Siemens, and comparing the NGS assay to the Lamprobe assay, they found 16 discrepant samples, as shown here. The Versant detected finally 90% correctly, and the NGS 100% correctly. On this table is shown that the Versant genotyping assay detected 1B falsely. The correct detection was the genotype 6, and that was all confirmed by Sanger sequencing. So the Sanger sequencing confirmed that all the NGS results were correct results. This is of concern, since if you would get the wrong information about the genotype, as I mentioned before, you would probably also choose the wrong regimen with the drugs, may fail, and may have additional cost for drug treatment. Interesting here is also that in Southeast Asia, here in Singapore, we got a prevalence of genotype 3 and followed by genotype 1B and 1A. You also have a lot of genotype 6 in this area, which is not really representative of the Western world. All right. If you look at the AASLD guidelines, which came out in 2016, in April or May timeframe this year, for the first time, there's a recommendation not only on genotyping, which has been in the guidelines before, but also on testing for resistance-associated variants. And it's a very comprehensive document. The source is given here, but if you look at the in a summary table, which is shown here, for the RAVs, which are detected by the Vela assay, which is shown here, it's about 130 in total. NS3 region, NS5A, and NS5B region. You can see that many of these RAVs we have recorded in our assay are also recommended by the AASLD guidelines for testing prior to treatment. And these RABs are shown here in marked in orange, and in more detail is shown here, specifically for the NS3 region and NS5A region, which are the most important regions when it comes to resistance-associated variant development. The NS5B region has a very, very high resistance to mutation, and we don't see a lot of, or at least not significant in NS5B regions, RABs which are clinically significant. So again, as you can see, most of the, actually all of the NS5A and the NS3 RABs which are recommended by the guidelines are covered by the Vela assay. The European guidelines which came out recently are calling for a less number of RABs, but basically giving you very similar information. Now let me switch to HIV. In our HIV assay, we're looking at 3 different regions which are relevant for treatment. One is the protease and RT region, and the other one, and this is a new addition compared to other assays which have been on the market before, we're also looking at the integrase region. Why is this? Integrase inhibitors have been on the market for quite some time. In many regions of the world have not been used very widely, and that is changing now, since especially in the United States, integrase inhibitors are more frequently used now. So we decided also to add integrase analysis into this assay. The number of amplicons we're generating is 2, and the targets, as I said, are protease, reverse transcriptase, and integrase. The schematic drawing of the HIV genome is shown here, and most of you, I guess, are familiar with this. And look at our own internal assay verification results, and as I mentioned, the assays developed here are all under ISO certified conditions, and we're following a very precise quality system. So what's shown here is data on the determination of the analytical sensitivity limit of detection shown here, and as you can see in the upper part of the table, when you look at 2,000 and 1,000 copies per milliliter, we have 100% detection in these experiments. Even if you go down to 500, you still have a 100% detection rate, right? As when you go here, samples with suboptimal median coverage, meaning 50 to 1,000, you see a little bit of a drop in at the level of 1,000 copies by 5%, and a higher one at 500. So we're recommending in our assay a threshold LOD of 1,000 copies per milliliter. You can actually lower, but the statistical significance or reliability then decreases. We also have seen in a crossover cross-contamination study based on 10 to the 6 copies per milliliter in 7 runs, we have seen no cross-contamination or carryover. And in our reproducibility study, we found in 17 runs 100% reproducibility. This is some data from alpha testing using this assay, and this is data from Professor Basson at the Thai Reference Center in Bangkok. They looked at 46 instrument runs here, and interesting here is that in the Southeast Asia region, we have a prevalence of the CRF01 AE recombinant HIV type, which is not highly prevalent in other areas of the world. But they looked at 676 samples in total, consecutive samples, found 620 CRF1s and 34 B, BC, and A1. And in the CRE1, they detected 96% correctly, and the B and BC and A1, the detection rate was 100%. In the meantime, we have also data from the United States, which is not shown here, based purely on subtype B, and the detection rate was nearly 100%, confirming this data here. In this study, they had 22 samples which were excluded due to low viral load or low sample quality. This laboratory has used the Siemens routine assay in the past. This assay has been withdrawn from the market recently. There's only one competitor assay left on the market now. The group looked at protease and reverse transcriptase used in Siemens routine versus the Vela assay, and they found 199 samples, 100% agreement, and for RT, they found 98% agreement. They also did QCMD testing, which passed with a 100% accuracy. So this data shows that the assay works very well. For the time being, this center is the assay, uses the assay on a routine basis. Some more data which we collected also in Southeast Asia over this year. It's 3,300 prospective samples tested in 2016. And when you look at the DMRs, drug resistance mutations, DRMs here, and the frequencies, this is the frequencies we're seeing here is very similar to what you would expect from using international databases. So again, we had a high prevalence of CRF1s here in this cohort, or not cohort, in this sample collection. And we detected more than 33,000 DRMs in total. What's shown here is a 10% frequency rate here for RT and protease, due to the fact that if you want to go down to 1%, the table would be totally cluttered. For the integrase region, which is less prevalent, where we see less DRMs happening, we're reporting at 1% level. So in summary, this is the features of the assay. 15 samples as one system control, similar or the same as for HCV. As I mentioned, sensitivity is at 1,000 copies per milliliter. Of course, we're concentrating on HIV-1 program. There are no cross-reactivities with a number of other viruses and human genomic DNA. The sample volume for this assay is 700 microliter, and we also, as for HCV, we're using plasma and serum, using the UDG system. Total assay time is 2.5 days, and the hands-on time is 2.5 to 3 hours. And the same as for HIV, HCV is fully automated. We have export option for fast A file. And in addition, and this is important, we have access to the Stanford HIV database for the detection of drug resistance mutation. Vela has taken, and this is my last slide, Vela has taken a license from Stanford University to get access to their HIV drug resistance database, which is the most important database currently worldwide. However, as I mentioned before, due to the export of the fast A file, the user can also blast their data against other databases, like the Rega database in Europe, the French database, or the Phenotigeno database. With this, thank you for your attention, and I'd be happy to take questions.
Thank you, Dr. Gerd Michel, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Dr. Michel, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type in your question in the box that appears on your screen and click the send button. We'll answer as many questions that we have time for. Let's get started. Our first question is, Dr. Michel, do you have other virus detection products in development or currently available? And if so, what are these products?
It's already on the market, and most of them are CE marked. We also recently got emergency approval from the FDA for our Zika virus assay. In addition to this, we have CMV, EBV, BKV, and other viruses which are relevant for testing in immunosuppressed patients. We also have chikungunya virus, we have dengue virus, and we have Zika virus, as I mentioned. These three viruses have become particularly important recently due to the spread of chikungunya and Zika virus worldwide, and especially in the United States, where we need to know the exact diagnosis and the treatment, for instance, for dengue, would be very different from chikungunya and Zika. So this, what we call the tropical panel, is currently also a very important product for us. So again, yeah, the question is, yes, we have a number of other virus assays on the market already, and we're continuously working on new assays like Triplex assay, which is very well advanced now for Zika, chikungunya, and dengue virus.
Dr. Michel, beyond saving on hands-on time, what benefits do you see from introducing automation to these workflows?
Well, as known from other automated systems in laboratory medicine, with the automation, you significantly reduce errors. And that is a huge advantage of automated systems in general, not only to the Vela system. So you reduce errors by laboratory personnel, and in addition to this, you're increasing precision and accuracy of your results. So reliability of your results in the laboratory provided to the clinician is increasing by this automation system very, very significantly. And of course, I mean, you're saving a lot of cost due to the automation. This is one of the reasons why many laboratories become highly automated today. And so it's a cost-saving factor in addition to the increasing quality.
Dr. Michel, you mentioned that the workflow is integratable with LIMS. How complex are the reports that are generated by this workflow?
Well, I mean, first of all, the reports we're generating are not complex. They're very easy to understand and very easy to read. They're very detailed, however, and when you talk about the connection to the laboratory information system in a given laboratory, our engineers will take a look at the specific LIMS in a given laboratory. So there is no general approach to connecting our system to LIMS, but we would look at this at solutions individually. This is not specific to Vela. This is an issue which we have been encountering in the diagnostic industry for a long time. And yeah, it requires a little bit of additional work, but usually due to the experience and the expertise we have, it usually works very well.
Thank you again, Dr. Michel. We're out of time for questions, but would you like to leave our audience with any final comments?
No, I mean, maybe a last comment on the introduction of NGS in molecular diagnostics, which is kind of a breakthrough in the analysis of viruses, and also, as we will see in our second talk on oncology. The information you're getting here is highly comprehensive and highly precise, and with the introduction of NGS in a routine, and we have these now for HCV and HIV on a routine basis. I should have mentioned that the HCV assay, which we have introduced recently, is already CE marked in Europe. It's been approved by TGA in Australia, the regulatory agency there, and is also being approved by the Singapore authorities. We're currently introducing HIV for CE marking. Validation studies are nearly completed, and so we'll have CE marking for HIV next year. We also have a pre-submission for HCV to the FDA. So this assay will probably be used more widely in the next coming month and certainly also next year. So we're looking forward to working with many collaborators, and if somebody has an idea about the collaboration or has a desire to collaborate with us, you can contact us directly and we'll get back to you. Thanks for the attention again.
Thank you again, Dr. Gerd Michel.
Our next speaker is Mr. Lou Welebob. Lou Welebob has served as the president of Vela Diagnostics since 2016. Prior to his current position at Vela, Mr. Welebob co-founded and served as the Senior Vice President of Research and Development and Technology at Vela Diagnostics in Singapore from 2011 to 2015. While at Roche Diagnostics Corporation, he served as the Vice President of Global Program Management and Business Development from 2008 to 2011, and as the Vice President of Marketing, Services, and Sales from 2005 to 2008. Mr. Welebob also held both scientific and commercial positions in several biotechnology companies and scientific research positions in several pharmaceutical companies. He received a BS in chemistry from Wilkes University and was a member of the National Research Honor Society. He completed graduate coursework in biochemistry while at the University of Scranton and graduate coursework in chemistry at Rutgers University. I will now turn the presentation over to Mr. Lou Welebob.
Thank you, Christy. Thank you very much for your introduction. And hello, everyone. Happy to be here on the LabRoots presentation, and would like to speak to you today with regarding a very exciting new development within our team, which is cell-free DNA detection in oncology. So particularly what we're going to look at today is the determination of somatic mutations using cell-free DNA. The first thing to understand is why are we so interested in this area? The first, basically, cancer deaths continue to increase. So the key to survival is early detection. So by 2030, the American Cancer Society estimates that 22 million new cases of cancer will appear per year, with 13 million deaths annually. Today, in 2016, there are approximately 14 million new cases with 8 million deaths annually. So the growth rate is expected to continue to rise over the course of the next several years. The other important thing to note is that at least half of the US cancers are diagnosed at stage 3 and stage 4. Okay, so this is late stage diagnosis, which is definitely affecting the survival rate. So the key thing is to come up with a molecular screening technique whereby you can detect the early onset of cancer in the earlier stages of the disease in order to provide an appropriate treatment as soon as possible, and thereby lessen the opportunity for metastasis and also to extend the life of the individual. So what have we done? We basically have come up with a system whereby we can detect mutations either using our next generation sequencing panels or our PCR assays from just 4 mL of blood or plasma specimen. Okay, so basically the way the system works is that 4 mL of blood or plasma are taken out of either a Struct tube or a K2 or K3 EDTA tube. They are placed into the Sentosa SX101 liquid handling robot for automated extraction of clinical samples. That extraction process takes about 3.5 hours. After the extraction is done, the robot is automatic. The robot can also be loaded with either a PCR assay, such as the Sentosa assay BRAF or KRAS PCR test, or the robot can be loaded with one of the next generation sequencing panels, such as the Sentosa SQ CRC panel, the SQ NSCLC panel, the melanoma panel, or our brand new Sentosa SQ Pan-Onco panel. Just as Gerd had mentioned in the prior presentation, both systems have automated reporting associated with them, and we also have the capability for the oncology assays to utilize a new tool for providing enhanced data analysis through a software called TheraKey, which is being offered through Vela Genomics as a service, okay, globally. Some of the key advantages of our system is the selective and high recovery efficiency of cell-free DNA fragment size of approximately 170 base pair, as confirmed by the bioanalyzer. The automated workflow with minimal hands-on requirements, the hands-on requirement about 15 to 20 minutes. Sample input volumes of 4 mils. Most other systems on the market actually require greater than 4 mil input volume, and the onboard extraction is 3 hours and 30 minutes. So with very little hands-on time, minimal sample amount, you can maximize the amount of data that you're getting out of one single sample of cell-free DNA from blood or plasma. So the Sentosa SX cell-free DNA extraction kit looks like this. Okay, so it's built for 4 runs with 8 preps per run. Okay, and just like the prior system for the HIV HCV that Gerd spoke about, the Oncology system runs 7 samples at a time with 1 system control. Okay, the kit consists of a protein precipitation buffer, a conditioning buffer, binding buffers, wash buffers, elution buffers, and then the beads and the system control. So as I had mentioned earlier, the Sentosa SX cell-free DNA extraction includes off-board hands-on time of only 15 to 20 minutes. We've extensively investigated the types of tubes to be used in this system. And the 3 types of tubes that we recommend for use in this system is the K2 EDTA tube, which is to be used for PCR. As we started to investigate the tubes, we noted that the K2 tube was not providing adequate results for next generation sequencing. So as many of you know, tubes have specific properties to them, which can interfere with certain types of detection or analysis. And so the K2 EDTA tube, while fine for PCR, did not work adequately for the next generation sequencing. Whereas the K3 EDTA tube worked fine for both PCR and next generation sequencing. The blood storage for 4 hours at room temperature, followed by plasma separation, is the recommendation for both the K2 or K3 EDTA tubes. For the plasma separation, we recommend centrifugation at 1900 G for 10 minutes at 4 degrees C, followed by plasma clearing, another centrifugation step at 1600 G for 10 minutes. Okay, and then after that, the plasma can be stored at -20 or -80 degrees C. For the sample type whole blood, we're recommending the Streck tube for that, which is stable up to 48 hours at room temperature before processing. For the sample input, volume of 4 mils. So again, we're transferring the cleared plasma to a 5 mil Eppendorf safe lock tube, adding 266 microliters of the protein purification buffer and vortexing for 15 seconds, centrifuging that at 6700 G for 10 minutes. Transferring the supernatant into a 5 mil oxygen screw cap tube and loading it onto the Sentosa SX101 liquid handling system with the 5 mil tube provided by Eppendorf. The SX101 will then add in the controls to the samples for the next generation sequencing assays. So this entire process I just described takes roughly about 15 to 20 minutes. Okay, once the plasma is on the deck of the robot, the first thing we do is we go through a binding procedure, which is automated by the SX101. Okay, followed by a second binding procedure on the SX101, followed by the washing procedure. Okay, and then finally the drying and elution procedure on the SX101. Okay, so through this procedure takes about 3.5 hours, as I had mentioned earlier, with a hands-on time of loading the robot for about 5 minutes with the desired consumables. The Sentosa SX101 robot is set up via computer prompted deck layout. Okay, so the computer will prompt you to what to put on the deck and where to place each component on the deck. Okay, so we have a consumables for the reservoir rack 3 at position 5, and that is where we place our system controls. And then all of the buffers are placed into a separate rack, okay, on the system. Okay, so after we do this, what does the data look like? So after we've provided the cell-free DNA preparation, we've then taken the prepared samples into our Sentosa SQ-CRC panel for looking into our Sentosa SQ-CRC panel. In the Sentosa SQ-CRC panel, what we did was we took negative plasma material and we spiked that negative plasma material with known quantities of cancer cell line. And this is the results that we obtained for 7 individual samples. Remember, there's always one control associated with the system as well. Okay, so for the sample number 2, we detected the V600E mutation and reported the variant frequency at 27.7%. Okay, whereas running side by side with a silica-based column extraction method, only wild type was detected. All right. Again, looking at samples 3, okay, 15%, and then all the way down on sample 4 to approximately 4% mutation. So very, very low, very low frequency mutations able to be detected with the system down to 4%. If you look at the bottom table describes the median coverage that was obtained from the sequencing system. So we're looking at an approximately median coverage of roughly around 2,000 to 3,000 reads for the detection of each one of these particular mutations. So after building some contrived samples, we actually obtained some matched plasma and FFP material from 2 commercial vendors, geneticists and Capital Biosciences, with various known stages of cancer being present. Okay, so in the first set of samples, we noticed that basically we're looking at the detection of wild type, and we detected wild type down to a level of approximately 1.49 and 1.11 nanograms per library prep. Okay, so very low input amount of material detecting wild type. The next sample was a stage 3 sample material with 2 mutations present, with a mutation present KRAS of T581. The plasma observed mutation was also the same as the FFP mutation, and again, note the amount of nanogram per library prep at 0.49. Likewise, for the following samples, we also detected a TP53 mutation, a PIK3CA mutation, and 2 additional KRAS mutations. Okay, again, down to a very low amount of material per library prep. So the panel that was used on this system was our colorectal cancer sample, which is targeting 11 clinically relevant genes in 113 hotspots. So when you're monitoring, say, a disease or a known affliction, using one of the smaller cancer hotspot panels is very prevalent amongst our customer base. Okay. So this is a smaller panel for CRC or even the smaller panel for NSCLC. These panels basically have about approximately 1,000x median coverage, all right, run 7 samples and 1 system control, can be used with 1 10 micron section of FFP material, or in this case, 4 mils of plasma, okay? So very high sensitivity, high reproducibility at 1,000x coverage for these panels, and these are for high prevalent mutations. Okay, so this is where you're not looking at, these are high prevalent mutations as determined out of the cosmic database. Okay, so the next panel we have is the Sentosa SQ melanoma panel. Again, 1,000x coverage, covering the high prevalent mutations associated with melanoma, as determined out of the cosmic database. Okay, so again, if you're monitoring a known case of melanoma over treatment, this is a desired panel to use. The next panel is our Sentosa SQ Pan Onco panel, and this panel contains high and low prevalence clinical mutations on 70 genes, including 3 whole genes. Okay, so this panel was designed in mind of looking for high and low prevalence clinical mutations. Unlike the other panel, which is designed more for known clinical mutations with high prevalence, this panel covers both low and high prevalent mutations. You notice on this panel, the median coverage is 500x with a minimal coverage about 300x, covering 67 hotspot genes, 3 whole genes, and 351 amplicons, utilizing 7 samples per run plus 1 system control. Okay, so this is a brand new product from Vela, and what we're doing with this product is we're actually offering early access program for cell-free DNA kit available for PCR or for next generation sequencing. The next steps in our progress toward the continued enhancement of this kit is to work with additional matched FFP and blood specimens in order to continue to drive down the level of detection, the level of mutation detection, and also to look at how low we can go with consistent detection. So I showed you examples of down to about 4% right now. And what we're doing in our current research is taking those down to under 4%, approaching 1% mutant allele frequency. From the initial results, that I've showed, you can see that it's possible to automate cell-free DNA isolation and library preparation on a single robotic workflow and detect somatic mutations from NGS at a very low frequency with a very low sample input amount. Okay. With that being said, I'd like to ask if there's any questions from the audience where I can continue to explain the system in a greater amount of detail.
Thank you, Lou, for that informative presentation. It's time for Q&A. We want to get right to your questions and input, so here's a reminder how to let us know what you have to say. Questions and comments can be submitted via the Q&A button in the lower left. We will try to get to all of you, but if not, we'll be sure to follow up after the broadcast. Let's get started. Our first question is, Lou, can your cell-free DNA kit be used with Illumina sequencing systems?
Thank you, Christy. Yes, the cell-free DNA kit can be utilized with systems beyond the Vela panels I showed. We have worked well with the Illumina platform in conjunction with both our FFP extraction system as well as our cell-free DNA extraction system.
Lou, have you tested the cfDNA workflow on the new PanOnco panel, and do you see similar results?
We're in the process of testing that today. The new PanOnco panel is also in early access at this time, but our goal is basically to have the cell-free DNA workflow work across the PanOnco panel. And what we're particularly looking at right now is to look at rare variants at low frequencies for use on that panel, and to see how we are able to detect those at the 500x median coverage.
Now, do you plan to offer gene fusions in your Onco panels in the future?
We see gene fusions as a very important prognostic, and so we do intend to include gene fusions for ALK, ROS, and RET into our panel in the future.
Lou, how does the early access program for the cell-free DNA reagents work? Do I need to purchase instrumentation, or will Vela make instruments available?
For the cell-free DNA, for the early access program for the cell-free DNA, we will work with the interested party to provide them with not only the kits but also access to our instrumentation for the extraction and for the detection, if that's what the desire is.
Let's see, we've got lots of great questions coming in. Let's see, okay. Lou, if an audience member submits a question, they say, if I'm not mistaken, you said about 4 to 5% sensitivity and mutation detection against WT background. Isn't it too low for liquid biopsy?
So 4% mutation detection. So our idea here is that we know that low frequency mutations are becoming increasingly important, okay? And so we feel that being able to provide low frequency detection to the community is important to monitor disease progression and to even potentially look at early disease progression. So we feel that although the guidelines today are showing higher, may show higher mutant allele frequencies as being clinically relevant, we also have been working with collaborators who tell us that we should basically be driving those mutant allele frequencies as low as possible because they are seeing clinical relevance of those in many of their patients today.
If there are no more questions, I would like to once again thank Mr. Lou Welebob for his presentation. Lou, do you have any final comments for our audience?
No, just that I think this technology really is the next generation. We've always talked about what the next generation sequencing was, what the next generation detection was, but here's a next generation pre-analytics that I think has wide-ranging opportunities to really improve the level of healthcare globally.
Thank you once again, Mr. Lou Welebob, and thank you to Vela Diagnostics for making this presentation possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us. We hope to see you again soon. Goodbye.